In this tutorial, I will cover some of the features that are useful for both drawing networks as well as analyzing networks. So I'm going to start by placing a few items on the screen, A, B, D, and then arrange them in different ways. For example, align evenly, align compact, two of the examples. This align compact is useful to have promoters, genes, and so forth. You can align them compact and they're arranged better. Now, the next feature that I want to show is, suppose that C and D represent a dimer. In other words, these are not separate molecules, these are a single molecule just drawn as two molecules. Then what you can do is you can select these items and group them using this button here, the grouping tool. When you group these items, what happens is you will notice that D has disappeared from the model. There is only B and C. C represents a dimer. C is essentially one item drawn as two different items. You can see they also move together. You individually control button. So this is a nice way of representing dimers. It is also useful in genetic networks where you may not want promoters genes to be drawn as separate items, but actually represent a single item. So you want the drawing to be as it is here, as three items. They represent just one item, not three separate items. And so grouping is again useful in that case. Now you see, only F exists, the other two have disappeared. Now, an additional visual descriptor you can add is on these connectors. You can select the connector, use this particular tool, the basic transformations tool, and then there is an option to show the center box. If you say yes to the center box, a small box will appear in the middle of these connectors. And then you can insert new descriptive icons here. So first, let me introduce you to a very useful feature in TinkerCell that allows you to decorate TinkerCell models in a very illustrative manner. And that is the ability to replace the graphics that represent each of the items. So I can select an item and then use this particular button here, the replace graphics. Alternatively, I can click the right button and then do replace graphics. And then I can look at this folder called the items, which contains several XML files. So for example, antibody. As you can see, the illustration, the graphical representation of the part has been changed to something that looks like an antibody. This does not convert the part into an antibody, simply changes the graphical representation. So similarly, you can change the graphical representation of this box to any of the items that are available. So perhaps gradation. Similarly, you can change the arrowheads using this arrowhead tool. And change their sizes. Coloring in TinkerCell is similar to any drawing program. So you can use this color option and color any part of any of the items. You can use different types of gradients, different colors, transparencies, and so forth. You can also change the edge color and width. F5 to fit all the items. If you have programming experience, You can add new functions to TinkerCell in C or Python code. So I will demonstrate a simple example of a function that is very useful. Suppose you have a promoter region and two molecules that affect the activity of that promoter region. So these would be two transcriptional regulators. Now the number of states that this promoter region can be in will become very large. The number of states will become very large as you get transcription factors that can bind to that region. So the number of reactions that can occur here can be enormous because of the different combinations you can get. Now there is a function that will automatically generate all the combinations of reactions. That is called this load full binding kinetics function. When you double click that function, it prints the stoichiometry that shows all the binding and unbinding events that happen when these two transcriptional factors bind to the promoter. When you highlight these reactions stoichiometry, you can see that these reactions represent all the binding and unbinding events that can possibly happen. Similarly, there is another useful function, which is this multi-step process. Suppose you have a promoter and a gene, or simply two molecular species. Now there is this elongation reaction that is specifically for items that are on the DNA, such as promoters, genes, and so forth. And there's just a regular reaction. Now if you want, by default, this particular reaction, this elongation reaction, is treated just as any other reaction. This promoter is not consumed and this gene is activated by this promoter. Now if you want to explicitly model this as separate steps, the separate intermediate stages that occur during elongation, then you can use this multi-step process and specify the number of steps you want, for example 5, and it will change the stoichiometry of this reaction. So now there are 5 intermediate steps going from A to B. As you can see, there's a cascade of movement going from one step. You can also do the same for any other reaction. Suppose there's a 3-step process. As you can see, it printed the stoichiometry here, and the stoichiometry will be altered here. Other useful functions are the print sequences. Several items such as promoters, genes, terminators, and so forth. Then you can double click on this print sequences and it will print the sequences of all these items. Of course, by default, they are all similar sequences. As I mentioned, if you have programming experience, you can add new functions to TinkerCell. A simple way to do that is a simple way to test an algorithm you might have in mind is to use this dynamic runtime C code or Python code. So to demonstrate a simple C function that will just select one of the items and move that item. So I can use the list of functions shown here to choose what functions I want. For example, I want to get an item, so I can use the find method. So I can say find and then object x equals tc find and say a. And then I can use another method called tc set position of that object x to let's say 200 200. So this found the item A and then moved it to 200 200. Let's move it 500 200. So you can see it shifted that item A. Similarly, I can select the item, tc select x, and it selected that item. I can also do this using Python functions. For example, x equals pytc dot find a and pytc dot set pos x to let's move it back to 200, 100, and let's do pytc dot x. So you see it moved A and selected it. Of course, these are very trivial examples, but there is a large number of functions available, which allows you to write your own functions that perform some intricate calculations as interface with Tinker visual interface. All of the functions that are shown in this box here use the same strategy, this C API, to interface with TinkerCell.